Okay, thank you for being here. So, since their seminal paper in 1975, King and Wilson highlighted how, at least in terms of nucleotide substitution in the coding region of the genome, chimp and human wouldn't have enough differences in terms of to justify the variability in the phenotype and the behavior between these two species. And they suggested to look at the non-coding regulatory portion of the genomes. So, finally, 45 years later, we are able, thanks to development of techniques like CHIP-seq and the decodification of the histone modification code, we're able to predict the location of the regulatory region of the genome, so enhancers and promoters. And that's what we did. So, in order to address these differences in the gene regulation between species, we looked at 6 different primate species in the liver tissue specifically, and we produced CHIP-seq data for 2 different histone modifications that allow to target enhancers and promoters in the genome in order to characterize tempo and mode for the evolution of gene regulation in primates. Also, we produced RNA-seq data for the same species in order to investigate the evolution of gene expression across the lineage. We tried then to correlate these 2 different typologies of data, and finally we wanted to characterize the features associated with the conservation and the evolution of these cis-regulatory elements, these CREs. So our taxon sampling included species from each one of the main lineages within primates, so mouse lemur and bush baby as the 2 most basal species, then a new world monkey like a marmoset, and old world monkey like the Rift macaque, and then human and chimp as representative of the great apes. We processed 3 or 4 samples for each one of the species, both males and females, and always with an healthy liver, so the cause of death was not associated to liver disease. So a key point of our experiment was to detect orthology between the CHIP-seq peak across the species. So we used a human-specific approach, and we took advantage of the multi-species alignment Ensembl in order to, starting from the human peaks, so the human regulatory regions, we looked for orthology in the other species. And each time that there would be an orthologous enhancer or promoter between human and the other species, we looked for differential CHIP-seq binding using DSEQ2, that is a software that is actually normally used for differential gene expression. So again, I want to remark that we didn't just compare the different peaks in the different species, but we did a differential expression analysis between the human peaks and their orthologous region in the other species. This allowed us to predict and detect more than 47,000 enhancers and promoters, so regulatory regions, that are found in human and have an orthologous in each one of the other 5 species of primates that we analyzed. And as you can probably appreciate in panel B, 62% of them are conserved. They mean they conserve their functionality as an answer or promoter in the other 5 species. So this is an example of this. You can see CHIP-seq binding pretty much in each one of the species. Conversely, less than 1% of them are instead human-specific, this, and less than 2% of them are age-specific, so enhancers that are a function shared between human and chimp, and whereas this same enhancer is not functional in the other species. 1% can seem like a small number, but we're actually talking about hundreds of enhancers and promoters that are shared only between human and chimp, and it's still a high number. So here we see in panel A and panel B, respectively, the differential CHIP-seq binding between human and the other species. So we compare the species in a pairwise mode. So human and chimp, human and rhesus, human and marmoset, and so on. And we did that for the CHIP-seq data, but also for the RNA-seq data. So here in the y-axis, we have the differential expression fraction for almost 11,000 genes that are annotated as orthologous between the 6 species. And here instead we have the differential CHIP-seq binding fraction. So enhancer showing a differential binding in comparison with human. And it's interesting because in both of the essays we see how the evolution of gene regulation and the evolution of gene expression, they both basically recapitulate the primate's phylogeny. So species that are more recently diverged from humans, so for example chimp and rhesus, have a lower number of differentially expressed genes and enhancers showing a differential functionality. And at the same time, species with earlier divergence, so bush baby and mouse lemur, have a higher number of differentially expressed genes and also a higher number of enhancers and promoters that are basically non-functional in these species and are instead functional in human. So, okay, our next step was, yes, we found that 60% of the human enhancers and promoters, so regulatory elements, are functional in each one of the other primate species, so they are conserved. So what are the features that are associated with this high degree of conservation? So first thing we want to look is where the genes associated to these specific enhancers and promoters. So we use the closed gene approach that is not 100% accurate, but it's still very good for this kind of analysis. And we look for the genes associated to our conserved and not conserved regulatory elements. And we found that enhancers associated with coding genes are significantly more conserved than enhancers associated to link RNAs or pseudogenes. This might be expected, but it was good that our results confirmed this. Then the second question was, what about the distance of an enhancer from the transcription source site? Is it associated with the degree of conservation of this specific enhancer? So here, in the x-axis, you can see quantiles of the distances of our enhancers from their associated genes. So they are basically beans containing a lot of enhancers. And in the y-axis instead, you can find the fraction of these enhancers that are conserved within each one of these beans. And so you can probably see how, as far as the species are recently diverged from human, so for example, chimp and rhesus macaque, the degree of conservation is basically uniform regardless the distance from the transcription source site. However, for species with an earlier divergence, so for example, the lemurs, the bush baby, after a threshold of 5 to 10 kb, you can see a dramatic decrease of the conservation of the enhancers associated to the gene. And this is showing us that as long as the enhancer is close to a gene, there is probably a sequence constraint that tend to conserve the functionality of the enhancer. But when you go farther away from the gene, probably the genome is more relaxed and the function of the enhancer tend to be more subject to turnover. Then also we wanted to look at the number of cell types where these enhancers are functional, because the fact that we predict an enhancer in liver doesn't necessarily mean that this enhancer is also a functional enhancer in other tissues, in blood, in brain, and so on. So we took advantage of publicly available data for DNase hypersensitivity. That is an assay that look for regions of the genome that are basically open and therefore functional for more than 200 cell types. In the X here you can find the quantiles again of the number of cell types, so beans containing several of our enhancers. And again, in the y-axis, we have instead a fraction of conserved enhancer within each one of these beans of cell types. And as you can easily see, there is a strong positive correlation between the number of cell types where an enhancer is functional and the likelihood that this enhancer is going to be conserved across more than one species. So on the other side of the coin, we can say, okay, if an enhancer is specific to a single or just a few cell types, it's more likely that it's going to be subject to turnover and lose functionality. Okay? And very similarly, here we performed a similar experiment. So we asked, okay, what about the number of transcription factors that bind to a specific enhancer? Are they associated to the fact that this enhancer is conserved or recently evolved or labile? And so we took advantage of ENCODE data for publicly available data for transcription factor binding H2 cells that are a subset of liver cells. And again, in the X, we have beans with the number of transcription factor binding our regulatory elements. And again, in the Y, we have the fraction of conserved enhancers within these beans. And again, we see that the number of transcription factor binding a specific enhancer is highly correlated to the fact that this enhancer is conserved across species. Finally, the last analysis that we performed was, okay, let's look specifically into some of these loci where we found both a human or ape-specific CHIP-seq binding, so human or ape-specific enhancer, basically, and also an associated differential expression of the associated genes, so genes that are upregulated in correspondence to an enhancer that is functional only in the specific species. So I'm going to show you just a couple of examples. So GERIT2 is a gene with a lot of literature. It's a very important essential gene involving liver, brain, neural tube development. And in our essay, we found a human-specific CHIP-seq peak, so a human-specific enhancer. And also the gene was upregulated in humans. And so we looked at the sequence, and we found that the CHIP-seq peak is in correspondence of a 1.9 kb insertion that is human-specific, as you can see, is absent in chimp. And this specific insertion is a transposable element, speaking about them, and specifically an SVA, that is a family, an omidyl-specific family of transposable elements. Now we're performing reporter assay experiments to validate the functionality of this specific insertion, so using a vector and a luciferase assay. And if that would be confirmed, that means that for these genes, humans have a novel specific enhancer that is derived from the exaptation of a transposable element. And this is a very important gene, so this result was kind of surprising. Sorry, I missed a slide. Okay, no worries. So we found this also in many other examples. For example, with green 3A, that is another very important gene, having a specific peak in this case, and at the same time, the gene being upregulated in apes. This gene is important because it's associated to schizophrenia. And again, we found a transposable element insertion in correspondence of the predictive enhancer. So since we found several cases like this, we tried to assess and quantify the contribution of transposable elements into the evolution of human and apes gene regulation. And we found, surprisingly, that 95% of our 57 human-specific enhancers overlap a transposable element. And most of these transposable elements are, in fact, SVAs, so an omidyl-specific family of transposable elements. And at the same time, if we look at apes, we found exactly the same thing, very similar. 84% of the ape-specific enhancers, they are 544, are transposable elements, but if we break them down, we see a more heterogeneous pattern in the different families of transposable elements, so SINE, line, LTR, SVA, and DNA-derived transposable elements. So just to close, so quickly wrap up, I showed how 60% of the human cis-regulatory elements are conserved, at least in terms of functionality, across primates, and more than 99% of the enhancers and promoters are active in human, and also in at least another primate. Variation in gene expression and gene regulation in primates, they both recapitulate the lineage phylogeny, and the conserved enhancer and promoters are mostly located within 10 kb from the transcription start site of the coding gene, and they are functional in multiple cell types. Finally, our approach allowed us to predict novel enhancers, species-specific, especially for human and apes, and most of them are associated to very important genes, and most of them are derived from the exaptation of transposable elements, evolving a functionality into a cis-regulatory element. And that's all. I wanted to thank you all, and my mentor, my collaborators, and the funding source.